Kelly Mistry, a friend, a colleague, a mentor, amazing scientist, on and on. Her more formal bio. That was the Kristen flavor. Kelly Mistry is a quantitative ecologist, currently working as a research scientist in the Quantitative Conservation Lab at SAFS with Dr. Sarah Covers. She earned an MS in the Quorum program here at UW in 2022, advised by Dr. Mark Sherrill. Her thesis work focused on comparing different modeling methods used to manage groundfish stocks, one of my favorites, in the Gulf of Alaska, including state space and spatiotemporal models. Kelly's current research is part of an ongoing project to monitor and manage an invasive species, the brown tree snake, in Guam. And outside the lab, Kelly is a trained facilitator and science communicator, as well as a fiber artist, I can attest, beautiful work, and poet. With that, I would like you to help me welcome Kelly to this stage. Would you like questions during or after?
Either one.
Okay.
I have an after slide, but if you have questions, by all means, you can ask in the middle.
Okay, I will let you know if I see questions online. Folks, you can use the Q&A option for that, and folks can use the traditional raise your hand model for the in-person room. With that, welcome Kelly Mistry.
Thank you all. Once again, thanks for coming for the last one of the quarter. Friday, last week of class. It's gonna be probably over soon for everybody. Okay, so I'm talking to you today about brown tree snakes and a modeling project. This is still in progress, so some of the work is still sort of being worked through, and that's where, if anyone has feedback on, I'll tell you when we get to that point. I would really appreciate anything that anyone has to add. So first, I also need to thank my collaborators with USGS, USDA, Fish and Wildlife. There's a lot of people involved in this project, directly and indirectly. And then I also have some funders to acknowledge and some other collaborators that are not directly co-authors, but they're still very valuable collaborators. So thanks to all of those folks. And now I'll just tell you how the presentation is going to go, and then we can get going. So first, we're going to talk about why we care about brown tree snakes in Guam, because they're pretty far away, but they are still pretty relevant to a lot of things. I'll go over some of the specific management challenges and the tools that I'm going to be using. And then the spoiler alert, the tool is a simulation model. And so I'll be describing that. There's three sort of big parts of it. There's individual-based population generating model. There's a simulated management actions operating within that model. And then there's a removals-based estimation model. And then I'm going to finish with a model demonstration of a simple scenario to sort of walk through the steps. So first, why do we care about brown tree snakes on Guam? This is where Guam is in this map here, the red dot, right in the middle of the South Pacific. And brown tree snakes are not native to Guam, spoilers. They are native to parts of Indonesia, New Guinea, and Australia, so kind of the same general area, but Guam did not originally have any snakes. And so they were accidentally introduced in late 1940s by the US military. I should have added US on there. Basically, after World War II, there was just a huge number of things and people being moved all over the South Pacific. And some of those things and people were being moved from places that had brown tree snakes to Guam. And there's been some great genetic studies done that indicate between 1 and 10 brown tree snakes could have produced the current population. So it didn't take very many of them to get across, but we're pretty confident that's when it occurred. And since they've been on Guam, so again, what is that, 80 years approximately. In that time, they have managed to basically eat all the birds. 10 out of 12 of the forest bird species are completely extirpated from the island, pretty much. Some of those none of them have fully gone extinct, but some of them are only living in captivity, or they're only in other islands in the Marianas chain, which Guam is the largest one in that chain. And then there's also some other impacts besides ecological damage. One of the biggest and still ongoing ones is that there are frequent and very expensive damage that brown tree snakes can do to the electrical infrastructure. They go up into the electrical lines and then they electrocute themselves and they die. That particular snake dies, but it also can take down the electrical grids. So from this year, or rather 2022 data, there were 227 incidents with an average of $500 per incident. So it's very expensive. and ongoing. And there's a lot of effort that's put into keeping them from getting up there, but it still happens. So anyways, so there's both ecological and other infrastructure impacts from these snakes. So let's talk about the management of them as it's currently sort of the big high point. So the biggest motivations for brown tree snake management is that there's an ongoing and very real risk of the brown tree snakes getting to other islands. Hawaii in particular, we're very concerned about, and there's a lot of effort and a lot of money and a lot of time and people and resources that goes into policing everything that leaves Guam to make sure that brown tree sticks are not going off on plane rides and getting to other places, or boat rides. So if we're managing outside of just interdiction, it will help with those interdiction efforts. And also with some of the birds, the two ones of note are Koko, the Guam rail, and Seahek, the Guam kingfisher. These are birds that are primarily being raised in captivity, and they have been for a long time. And so if we want to do reintroductions with them, there's kind of an urgency that we sort of need to be doing it probably pretty soon for some of the species. And they basically, again, this was a snake naive island, so these birds had no protections against snakes when they arrived, and would probably need to severely suppress any populations or fully eradicate in order for reintroduction to be successful. So some of the big challenges, these guys are arboreal, so they go up in the trees, and they're nocturnal, so they're very difficult to find, basically. And there's also some challenges. Some of our management methods, which I'll talk about more later on, are prey-based, or they're bait-based, and these snakes actually change what kind of prey they prefer. So some of our methods only work for some portions of the population. So that's pretty challenging. There's also uncertainty about sort of the large scale population throughout the island. And while we have done small scale eradication experiments, we haven't done anything that's larger than, I think 50 hectares is the largest that we've currently done, which is not that big relative to Guam. And so there's just uncertainty about what a large scale management action would even look like, let alone how it would work. So that's all of the problems. So one of the tools that we as managers have to address areas when there's a lot of high uncertainty like that is we can use what's called dynamic management. The basic premise of dynamic management is you create a management plan, you implement that plan. Part of the plan is to gather or collect data in some way, and then you analyze that in order to update your assumptions or fill any knowledge gaps about the system or the individual populations, things like that. And then you use that knowledge to create the next plan. So it's a pretty basic concept, but it's an important one. So in this system, things that we could be learning about are the total abundance or relative density of the snakes in some area, the size distribution, because I'll talk a lot about size later on, but basically there's management implications for different size distributions. We could focus on some demographic rates. Some of their demographics are very difficult to study. Reproduction in particular has been really challenging. And then individual growth is also pretty important for some of our management objectives. And then we also could look at more encounter and mortality rates from our methods, see if those things are changing over time, if they're becoming acclimated to any particular method, that kind of thing. So these are things that we could learn about. All right, and so for this particular model, and also it's just a good tool for dynamic management in general, is simulation modeling. So simulation modeling is a very cost-effective way to test a lot of scenarios in a relatively short amount of time. I mean, it doesn't feel like a short amount of time when you're running a lot of replicates. It's taking weeks, but if you're comparing it to on-the-ground experiments, it's very fast. You can use it to identify sort of critical knowledge gaps. In this kind of a system where we have a fair amount of knowledge gaps, we probably don't want to try to fill all of them. So we want to figure out which ones are going to actually have the most impact on our outcomes, our management outcomes. So here in a simulation model, you can kind of pick and choose which ones you're going to work with. You can also explore all kinds of assumptions about the population, about their response to your management. And then you can use simulations to calculate probabilities of reaching, for example, a given goal with some level of uncertainty associated. And then you can use that to make decisions. So let's talk about this particular simulation model. So the goals of this model is it's a tool for optimizing BTS eradication given trade-offs. And the two primary goals that we're always going to be trading off against each other with this kind of management is maximizing the number of snakes that we remove from the area and minimizing our costs. There might be some other goals involved depending on the scenario, but these two are always going to be like the main ones that we're trading off against each other. And the primary users, what we're expecting is management decision makers. And then there are some sort of research applications as well. You could play with the population itself and sort of experiment with that. So there might be some applications there too. All right, so I'm going to describe sort of the big picture model structure, and then I'll go into a lot of detail about the rest. By the way, these pictures are all ones I took on Guam. They're mostly random locations, but they're pretty. Okay, so in the simulation, I have a generating model that is individual based. And I take that model through time, and the snakes grow, they reproduce, the individuals grow, they reproduce, and there's also natural mortality that's operating for however long I want. And then I can put management actions onto that population, and the 4 management actions, I'll describe these in more detail later, but basically there's 4 that we currently use. Live trapping with live mice as bait, visual surveys where people literally go out with flashlights or headlamps and look for them by hand. And then there's 2 methods that use toxic baits, which are also most of the time mice. One of them is aerial delivery system, which is coming out of a helicopter and can cover large areas. And then the other one is toxic bait tubes, which are something that people deploy on the ground. And in the simulation, so I apply these management actions. It produces individuals that either have survived or have died from those methods. And 2 of the methods, the live trapping and visual survey, those 2 actually produce actual snakes. We have the bodies of the snakes that are removed, and so we can get information from those. As opposed to the toxic bait methods, which don't, because they decompose very quickly. They might take the bait and then go a little ways before they actually die. It's just, we can't really get information from those methods. So from the 2 that we do get information from, we can get monitoring information and use that to estimate the population. And this is a size-based estimation model. The version I'm currently running has 4 size classes, small, medium, large, and extra large. Yeah, I'll be describing it in a lot of detail shortly. Okay, so first we'll talk about the individual-based generating model. So there's only one state variable in this model, it's the snakes, and each individual snake has an individual ID. They're randomly assigned sex, and there's also a randomly assigned growth quantile, which I'll describe what that means in a minute. They have a starting size, which the size for I'm going to use this a lot. The metric for size is SVL, snout vent length. It's a standardized measurement for reptiles and amphibians. And also reproductive probability is an individual characteristic, and that's based on their size. And we'll describe that one too in a bit. And then I have 2 sort of levels of timescales. There's a daily, or in this case, it's nightly timescale, and nightly they have individual growth, natural mortality occurs, and eradication activities can occur. And then at a quarterly scale, so every 3 months, reproduction occurs. And one thing you may or may not have noticed is there is no spatial element explicit in this model. And that's partially because I was trying to conceptualize an IBM that had this level of detail, and movement was exhausting to even think about. So at some point in the future, it might be worth someone's time to do that. But I decided for this purposes of this model that spatial scale was going to be implicit. And basically, there's 2 main things that are controlling it. There's a density function for the initial population size, and then there's a density dependence reproduction parameter that also has a density parameter in it. So come back to that later. Okay, so I'm just going to step you through the sort of steps of the IBM. So at the beginning of each quarter, the model chooses which females are going to reproduce. The females that are going to reproduce then produce offspring to give us a total number of new offspring, which are then assigned IDs. The sizes is drawn from a uniform distribution of between 250 and 350 millimeters, which is, so hatching size essentially. And then a randomly assigned sex. And then for each night in the quarter, these operations occur. So first, natural mortality is applied. Do they die from that? If yes, they're removed from the population. If no, then they go on and the model checks to see if they've reproduced in this quarter. And if they do reproduce, then they go straight into the surviving population. And the rationale for this is that one of the main challenges with brown tree snakes is we have found very few females that have eggs. And we're pretty sure that it's because they probably just kind of stop moving once they're procreating. And so they're probably also not eating very much. And this is an assumption that may not be true, but for the purposes of this model, I chose to assume that they weren't eating. And if they're not eating, then they're not going to grow. So the next step, if the individual didn't reproduce in that quarter, is the do they grow step. And if yes, then there's a function for how much. And if no, either way, actually, if eradication is occurring, then the next step is, are they going to die from the eradication methods that are on that day? And if yes, they're removed from the population. And if no, then they're added to that surviving population. And then the final step is after all the nights for the quarter have gone through, the offspring and the surviving population is combined, and that's the starting population for the next quarter. All right, so now we're going to talk about the submodel functions. So for individual growth, there is basically the question of, does growth occur as a Bernoulli draw? And right now, this parameter is a constant. I just wanted a small amount of stochasticity, basically. I did originally want this to be a density dependent parameter, and it wasn't the first way I was trying, it wasn't working very well. And so basically with my collaborators, we decided that it wasn't important enough to do right now, because a lot of the eradication scenarios we're talking about are probably going to be relatively short term. And so the amount of growth that might be density dependent, it's probably not going to have a lot of impact. So that's something that could be played with in the future though. All right, so if growth does occur, then how much growth? And so this is when the growth quantile that I mentioned comes in. So basically there was some really great work done by Bjorn Lardner and group that's unpublished right now. It will be in the future. And they looked at a population in a study population on Guam that had a lot of experiments done over the years. And they've got some really nice mark recapture data for snakes where they have repeated growth measurements for individuals. And so they were able to use this data set to sort of create growth profiles for the individual variation that they were seeing. And the model that worked best with their data had sex-based intercept, sex and this growth quantile. They actually produced, I think it was 7 growth quantiles originally. And I took the top 5 because the lower 2 weren't reaching reproductive size, and I wanted all of my snakes to potentially reproduce. So anyways, yeah, so the intercept and the slope is coming directly from their work. And this graph is, if there's no stochasticity, this is what the growth looks like for each of those quantiles. So they're randomly assigned at birth, which one they're going to have throughout their life. All right, so now reproduction, which has a couple different steps in it. So the first question, once again, does reproduction occur? And there's 2 parts to this. There's the individual reproductive probability, which is size-based, and then there's density dependence, which is the same for all the individuals in the population. And this is also a Bernoulli draw with these 2 multiplied together. And then if reproduction occurs, how many offspring are produced? And this is a Poisson draw if it occurs. And this is based on work from Levin et al. And Nafis et al., the two related studies. And they were able to do a genetic study of snakes in the closed population and in the study population. And I think they were able to, I don't remember now offhand how many individuals. It wasn't a huge sample, but they were able to trace offspring to mothers. And so from that, over I think it was an 18-month period. And so from that, I was able to get an average clutch size. And so that's what this lambda is, it's 2.2. This is also not size-based, which in reality, probably larger snakes. There's some evidence that larger snakes would produce more surviving juveniles, but I didn't have good data for that. And so I just chose to make it a single value. So this is another thing that might be experimented with in the future. Yeah.
Sorry, what in that parameter P for density dependence? I guess I usually think of density dependence as a function of.
Yes. Yes, it's not. So I'm calling it density dependence because it's operating that way, but it's not being created in the same way that it's not truly density dependence. So it's basically capping the population at what we've estimated from previous work. I'm going to describe it in 2 slides of what that's actually being produced by. But yeah, it's not truly density dependence in the actual sense. I just couldn't think of a better term. Okay, but first we'll talk about the size-based, how that's set up. So this is basically a Gaussian cumulative distribution, and it's based on, this is from Rhoda Savage 2007, which a lot of people have used, and basically they produced a 90% confidence interval for when female snakes transition to being reproductively active. And I just used that to create a Gaussian cumulative distribution. And I used it for both sexes because it was pretty close for the males. So the values weren't that different. So that's that part of it. And then density dependence. So this is a half normal distribution. And I've set this K. So this is a static value based on the area size. It's 60 snakes times the area. And I've assumed that that's a 97.5th percentile of this distribution. And it's not truly density dependence, but it's operating sort of well enough to cap the population. This is definitely a parameter that should be experimented with to sort of figure out exactly how much impact it's going to have on your eradication strategies, because it's relatively uncertain. Like, it is based on past research and literature review, but a lot of those studies were being done in the 90s and early 2000s when we're pretty sure the population was very high. It was probably over carrying capacity. Like, the conditions of the snakes they were very small and kind of skinny and not very, like, kind of sick looking. So this is like half that. Basically they were assuming a fair amount in order to come up with a figure. And so this is something that should definitely be revisited. But for now, that's what K is in this model. And the final piece of the IBM is natural mortality, which is pretty straightforward. It's just a static value, which is based on these four size classes. And there's not been much research on natural mortality because mostly we're trying to kill them ourselves. So any other mortality is just a bonus. But I did have, basically there was one study where there was a sort of a time frame, and they were tracking them, and they recorded how many had died before they really started their experiment. And so I used that to produce some static values. So all right, so any questions before I go on to the next step? Any questions on this part? Yeah.
I was wondering, do they have any predators?
Not on Guam, not really. Well, I mean, that's not entirely true. So at the larger size classes, no, really they don't, except for cars. That's actually like the most cars and weed whackers and stuff. Like, that's the main predators. Yeah, actually, genuinely, yeah. But for the small snakes, there are some predators. There's but not very much. Like, there's feral cats and feral dogs and stuff like that. So I actually artificially inflated my small value here because there weren't really very many small animals in that size class, and I assumed there was going to be a little bit more mortality. So, but yeah, there's not much. All right, so now we're going to talk about the management actions. So first we'll talk about what the actual actions are in real life, and then I'll talk about how I am applying them here. So first, there's visual surveys, which this is a group of USGS field techs going out to search for snakes. It is literally by hand looking with headlamps. Then there is live trapping, which is with live mice. So you have to maintain the mice populations, but it's a bit more passive in terms of the traps can be out all night. Visual surveys occur for 4 hours per night. And then there's the toxic bait methods, which these are primarily being used with dead neonatal mice, and they literally strap a little tablet of acetaminophen to them, which the nice thing is acetaminophen is pretty much only fatal to snakes in this system. There's really not much else that's going to be affected by it, so we don't have to worry too much about introducing toxins that's going to be harmful to other species. So that part's good. And then the two methods that this is delivered with is, again, the ADS in the helicopter or bait tubes on the ground. And these three methods all have size-based mortality because they're mice baits, various forms of mice baits. And then these two methods are the only two, this visual survey and live trapping, are the only two that produce removals data. So the toxic baits don't typically. Okay, so in the model itself, there's sort of two steps to this. The first step is, because this is a spatially implicit model, I first have to decide how much of the population is going to be affected by any given method. And it's a pretty simple, basically, I just calculate how much spatial coverage that method has, and there's some standard, there's been good research about that in terms of if there's a transect, how far do certain methods sort of have influence from that transect. And similarly with ADS, we know sort of the radius of drops, how much influence there might be. And so I can use that combined with effort to get a spatial coverage estimate. And then I literally just multiply that by the population. And so it is assuming kind of a uniform distribution, like there's some assumptions built in here that are probably violated in real life, but for now, this is good enough. And then once that susceptible population is set aside, it's for each method, then the next step is to figure out if the snakes are going to die, if the individual is going to die. And it's another Bernoulli draw, and this variable, the D, this D variable is different for each method. So I'm going to step through each of those methods. So the first one is nice because Stacy Ambergy, who many people here know, did some work for me that I can just pull her results from. So this is a post-area distribution of encounter probability for visual survey from a mark recapture experiment in the closed population, the study population in Guam. And you can see these are very low numbers for the encounter probability nightly. But also, I don't know how well you can see it, but basically she used the same size classes that I've been using, but all of them for visual survey are kind of overlapping. So it's basically kind of the same encounter probability regardless of size for visual survey. For live trapping, it's quite different. So this is, again, I don't know if you can actually see it, but it's basically as the snake gets bigger, the encounter probability gets bigger. So that lowest amount, that's for small snakes. You really never get small snakes in the traps, or very rarely. And then as the snake gets bigger, the likelihood goes up, but it's still very low. So, and for my model right now, I'm just using the mean value for each of these size classes. In the future, like you could use the whole distribution or, have some level of stochasticity in there from it. But for right now, it's just the mean value. Okay, and then the mortality for toxic bait methods has a couple pieces to it. So there's the baseline mortality, which this particular figure is from NAFIS et al. 2022 study, but there were several other ADS studies that had similar values. So it's, and there's been sort of other consultation that this is a probably decent value. And then this baseline is multiplied by a size-based susceptibility. So there's two parts to that. The first part is when small snakes are transitioning from lizards, which is primarily what they're eating when they're really little, to mammals. And then at the other end of the size range, there's sort of a decline. Mostly it's probably about bait attractiveness because the toxic baits are on like little tiny baby mice. And so for a large enough snake, that's just not really that appetizing. So that's probably why they're less likely to eat it. There is also, in some rare cases, the standard dose a single dose might not be lethal for the really big snakes, but it's probably more about attractiveness. So anyways, so there's two pieces to that. This is what the whole distribution for that variable looks like, with the top level being the 0.43. This first part is basically I did a Gaussian CDF with some limits, and those limits are based on literature review, but also sort of just expert consultation about when those transitions kind of happen. And then I have a Gaussian decay function for the upper size decline. And this one was also put together. The scale parameter for this was basically just the expert consultation of figuring out what people think would be the upper limits of when they would be interested in the baits. Okay, so all of that together, I'll just walk you through how that operates in the model. So I have this simulated population. Snakes are somewhere in an area. I have a couple eradication methods that are occurring for some of that area. So let's say method 1 is operating on 50% of the population. Let's say method 2 is like maybe 60%, which overlapping methods is possible in the code. And then let's say this goes on for a quarter, which is my typical time scale. And then the results that I'm getting out of it are the effort, so the number of days for each method that each method was used. And I have a standard number of hours for each of those, for each method. And then the other thing is the number of removals. So how many of each size class each method removes in a quarter. Okay, so before we go into the removals model, any questions about this part? Is it anything online, by the way? Okay, cool. Okay, cool. Well, let's talk about the removals-based estimation model. And this is the part that is still it is running, it is working, but there's still some pieces that I'm continuing to work on. So if anybody has ideas here, that would be cool. All right, so first, we're just going to sort of do an overview of what all the pieces are going in here. So this is from Link et al. 2018 removal model, and there are primary sampling periods. Within those primary sampling periods, we have secondary sampling instances. And then for my model, I also have different methods, multiple methods going on, and I have multiple size classes. And again, this is the size class sizes in SVL range. So what that looks like is I have, let's say there's 2 primary sampling periods. Within that primary sampling, I have, up to however many, most of the time this is going to be quarters for the primary sampling periods for my model. And the secondary sampling is days or nights in this case. And then within each night, there could be multiple methods, and also it's all of the size classes. So we're estimating based on the removals from that day, the population on that day for that size. And then there's a separate process that is estimating between sampling periods. So the assumption is the primary sampling period is closed. There's no, the only changes to the population is from the removals that we're doing. So that one thing about that is that assumption does require it to be relatively short timeframes for the primary sampling methods and our periods. And then also there can't be, like, say, ADS occurring in that same time period in that same area, would be violating that assumption. So, and then in between primary sampling periods, the population can change. It can be any kind of length of time. We could apply other methods during that time, all that. Okay. All right, so first we're gonna talk about oh yeah. Yeah.
So in your generating model, you had daily survival. And now here you have between primary periods, that's when your change is happening.
Yes.
Is there a disconnect? And so
Yes.
then
Yes, so the generating model okay. I was gonna talk about both of this then, but I can talk about it now.
sorry.
No, it's fine. I can talk about it now. So the first version of this, I was trying to map pretty directly onto the generating model processes. And so I had survival for the size classes, I had reproduction occurring, and I also had like growth of snakes that were transitioning from one size class to the next one up, that kind of thing. And basically this method just does not produce, even when I artificially inflated the encounter rates to see if I could get it to work, it just doesn't produce enough data. And so it can't estimate all of those parameters. And so I've had to simplify quite a bit so that it's not directly matching. I can still, because it's an individual based model, I can still get like true growth of the size classes between quarters, between primary periods. I can get that. And so I can compare reality to the estimation model, but it's not really giving a lot of information, unfortunately, about the population. And yeah, it's not directly mapping on. So that is one of the problems. Okay, so first we'll just go through how N is estimated within primary periods. So I have this fun equation, and basically Y is removals. And this is, so it's sort of every single day, so every I is, and also every method on I, in I, because sometimes multiple methods are going on, it's updating N for each one of those. It's for every, throughout the period. The other part about removals, of course, is that we have this encounter probability, and the encounter probability is estimated using our effort for that day, for that method. And then these two parameters, the intercept and slope here, are drawn from a uniform distribution. priors are drawn from uniform distribution. And I'm having some issues with those two, but it's performing well enough. So we've kind of moved on from those issues right now. And then the other prior that's important here is I have an initial N, so the starting population, first day of the first primary period. And basically the current method is that I sum all of the removals, like, so the absolute base amount of the population is the number that have actually been taken out for the whole time period. And then I have another like sort of some amount added to that. And this one is also problematic, which I'll come back to. But that's what it's currently set up as. Okay, so that's within the primary period. And then between primary periods, I have a Poisson draw with the remaining population. So this is after we've updated N every single time within a primary period. The final time is the remaining population. And then we take that and we multiply it by a transition matrix. And my transition matrix has simplified. So at this point, I only have size class based growth. And you may notice they are not related to each other in any way. So basically this population is estimating how the small size class grows through time, how medium grows through time, and they do not have a relationship to each other, even though of course they do very much so. But that's currently what we're doing. So, and this is gamma priors for these growths, and it's just going to the number of days between. So the R is a daily growth rate. Yeah, okay, so that's the estimation model. The next step is the model demonstration. But any questions before we go to that? I think I'm still doing okay on time. Yeah.
Sorry, just going back to the previous slide, you had the like your buffer of
Mm-hmm.
the top right,
Mm-hmm.
right? That's your buffer of your
Unknown starting population. Yeah.
Okay.
Yeah.
And then for the removals part,
Yeah.
too, because there's like the unobserved removals as well.
Well, that's the thing. So this requires the assumption of this requires there are not unobserved removals during a sampling period. So otherwise, yeah, this wouldn't work. So
then I missed it. So then this is just for the two methods.
yes, this is just live trapping and visual survey. Yeah, are the only two methods.
Because I imagine you can also add like a very small buffer kind of for the other methods.
Maybe, yeah. I'm having enough trouble with this one as is that I haven't explored that yet, but yeah, there might be a way to do it. It's a deceptively simple model that is difficult to get to work. There's two other people in this room who are also working with similar model and having problems with it. So, but yeah, that's an interesting idea though, to add just incremental amounts. Okay, let's keep going for now. Okay, so I'm gonna walk through a very simple sort of demonstration just to give a whole picture of how you might use this in actual decision making. So my model scenario is I have a 50-hectare target area. That, by the way, that was chosen mostly because that's kind of the smallest amount of space that an ADS would ever be deployed for. It's just not cost effective at smaller scales. So you've got 50-hectare area. I'm assuming this is a closed population, which, by the way, there is such a thing as snake-proof fencing. They've put a lot of research into figuring out how to snake-proof a fence. Basically, it's the one that's most often used is chain link, which is like a bulge that is a significant enough angle that they drop off. So anyways, so we're assuming this is a closed population. And there's also parts of Guam that you wouldn't be able to put transects on. For this scenario, I'm assuming the entire thing is you can do transects throughout. All right, and I run it for a single year. And the only amount of uncertainty that I'm adding is I have a stochasticity on the starting population. So there's an average density of 50 snakes, but I have normal distribution draws that are determining the actual starting population. So I have two scenarios. So scenario one is kind of like throwing everything we've got at them, all in full effort throughout the time frame. And then scenario two is we're primarily using ADS with just a little bit of visual survey to get some removal data in order to do the estimation. And the point of these two is not so much that either in of themselves is super realistic, but more I just wanted very contrasting scenarios to compare. So for each quarter, this is what the method applications look like. So I have the first 6 weeks that are my primary periods, essentially, where I have visual survey and trap only going on. The rest of the quarter, bait tubes and ADS. There's 1 round of ADS, which is 3 applications over one per week. And ADS is only occurring in the second and third quarters also. It's not every quarter, but the others are every quarter. And then the second scenario, ADS first, has 2 rounds of ADS with just visual survey. And then for the first quarter, it's just visual survey as like to set a baseline. And then second through fourth have ADS. All right, so let's look at some basic results. So you can see the stochasticity in the starting population. This is total population, the mean estimated total populations. And the all-in approach, they both sort of start out in similar, as you would expect, in similar places. ADS first does appear to be producing lower total populations. And also that amount of uncertainty at the beginning doesn't seem to be all that important, like the populations are coalescing more as it's going on. So that's useful information. And then let's look at the estimation part. So the all-in method is estimating that there's some significant amount of population, and ADS first is estimating there's like no stakes out there. Why are we even doing this? So let's compare to the reality of the simulated population. So the all-in approach did a decent job of estimating. ADS first did a very bad job estimating. But number one, estimation is not actually the objective, probably. The actual objective is getting the population down. And so estimation is useful because we want to be able to know what the population is doing in order to adjust our methods, but this might not be a bad outcome for the ADS first. So just putting that out there, especially if this doesn't include the size distribution. This is just the total. And so the next thing to look at is the size distributions. So the top row is going to be the all-in approach, and the second is ADS. And this has 95% confidence intervals. It's all the replicates, so that's why you see multiple values. And once again, like the all-in approach is definitely doing a better job of estimating, which is probably just because there was a lot more effort, and so there was more removals data to work with. The ADS first probably had a lot of zeros, and also not very much at all to work with, not very many days of effort. And so it's just doing a terrible job. But if your outcome that you're looking for is to have particularly the upper 3 size classes that are going to be threats to, say, birds, whereas the small size class, not so much. Like the diet transition is kind of between small and medium. So most of the small size class is not going to be, and also that's about where reproduction is occurring too. So the small size class is not, most of them are not reproducing yet, and most of them are probably not eating birds. So like, this might not be a bad outcome for either of these scenarios. Both of these scenarios might still be objectives that could work, or they might meet your objectives. But if you actually think that the entire population is very, very low, because that's what your estimation model is telling you from the ADS first approach, then you might not be appropriately responding when those small snakes start growing. Anyways, so the way you actually make decisions with this kind of a model is you have some kind of trade-off, of course, because remember we started out with the 2 objectives of maximize number of snakes removed and minimize cost. And there's a couple different things that there's a lot of different things you could do. 2 options are you can look at the eradication impact. In this instance, I chose total population in the final quarter, estimated population in the final quarter, or no, this is true population in the final quarter from the simulation model. And that's on the y-axis, with the x-axis is cost. And this one is a clear winner. If this is your metrics, you would definitely want to go with the ADS first approach because it's less expensive and it produces a smaller population at the end. But if you want to know what your estimation model is doing and how much error it's producing, so this is RMSE on the y-axis, and of course we want lower RMSE and lower cost. But depending on your objectives, either of these scenarios could work. If you valued the error more, then you would want to go with having lower error, then you'll want to go with ADS first, or with all in. And if you valued cost, then you would want to go with ADS first. In a real scenario, of course, we have many more strategies that we're evaluating. It's not going to be just 2, but this is just a demonstration of how you might use this. And finally, I do want to point out neither of these was actually dynamic management, but you can use the model to do dynamic management. So as an example, if you took the ADS first approach and instead had some kind of a target that you were working with, And let's say you paused after the third quarter of rounds and you did your estimation and all of that. And you had, let's say, I have a fake threshold here of 5 total snakes that you're being estimated, and the upper 3 size classes are all meeting that. We know that ADS is really not effective for small snakes at all. And so that final quarter of ADS was wasted money in the ADS first scenario. And so instead, you might want to switch to, say, visual survey, which is the only thing that's really effective for small snakes. So this is an example of a thing you would do. I didn't have time to actually do it, but anyways. So some of this we've already covered, but I do want to talk about sort of some of the big challenges and things that I'm still going to be doing. I have another 4 months that I'm having this project, so I don't know if I'll get to all of this, but hopefully. So one of the big things is that it is really difficult for this estimation model when there's very few removals, when it's just a lot of zeros. And since the purpose of this model is for eradication, that's kind of a problem. So one of the things that I am going to try next is adding a zero inflation parameter in there for the between primary period sampling or estimation rather. The other thing is that initial N for all the size classes is actually quite influential. When I underestimated it deliberately, it did a very bad job of estimating. So it can't really go up above. So I'm going to experiment with some other priors for that. One of the ones that we're probably going to try out is a Jeffries prior. And then the other thing is the single size class, having just one size class growth parameter would probably be problematic, particularly if there was ADS applications occurring between some sampling periods and not between others, because ADS it's going to be a large mortality probably just because of the scale that you have to do if you're going to do ADS. And so, right, for this, I had the same growth, but I would probably be better to at least have 2, one that's a growth rate when there's not major eradication efforts going on, and one when there is. So that's a thing that I might add in the future, and hopefully it wouldn't be too many parameters. It might be. So. Okay, so we have 7 minutes or something like that for other questions or ideas or whatever. Anything you guys want to add?
I'll monitor online for questions as people are typing, but we'll start with the room. Anything?
Thank you. It was interesting. I was wondering if you have put any thought into like longer term simulations. So like you could assume that whichever strategy you use for the eradication, you're unlikely to get it done in one of the sites. So
yeah.
And like at the end of those eradication efforts, like whoever's left is going to then make more snakes.
Yes.
Supposedly. So like how many cycles of this do you think one could need to get rid of
I think it's a great question. Certainly, like one of the things that I would love to experiment with is how different size distributions bounce back from eradication. So like after eradication, if it's only small snakes left, how quickly do they come back and get into reproducing age and, you know, raise the population and stuff. Or if we miss a few extra large snakes, you know, is that is missing like 1 or 2 of them enough to have it be quickly bouncing back, that kind of thing. So that's, yeah, I think that would be really interesting to try out. The another limit that I didn't mention of the estimation model is it takes a while to run. I think the current version that I'm running for a 50 hectare area was taking about 5 hours. So yeah, so I'm a little bit having to choose, you know, what things I'm going to experiment with. But I would like to do that because, yeah, I think it will probably take multiple rounds for sure. And the question is like, what is the optimal, depending on the specific context, it would be a little different, but what's the optimal amount of time between those rounds so that you don't let it get like kind of at a runaway stage, but also they're big enough that your methods will work. Because doing just visual survey for weeks and weeks and weeks with like hundreds of people, like that's never going to happen. It's just not feasible. So yeah, it's definitely a thing we need to experiment with and figure out.
John W. sent a link, which I've now emailed to you, so
Mm.
you don't have to keep track.
Okay, thanks.
Apparently they're using snake sniffing dogs in Florida for plague.
Yes. Okay, I didn't talk about snake sniffing dogs, but yes, they are using them in Guam. The reason, I mean, I would like to see that link actually, but the reason they're not using them, I didn't mention them here, is because they actually have tried. And basically these dogs are relatively small, the ones that they've got trained, and they use them for interdiction methods. So they're used for the airplanes and the shipments and all that. And they have quite a kennel. USDA has quite a kennel down there, and they've got a really great program. But they have tried taking them out into the jungle, and it's just not really a safe environment for the dogs. So basically, and also there's so many snakes around, they're just kind of saturated. They're like, oh, there was a snake here. I was like, okay, well, where, there might be some applications where it would be useful, but generally speaking, it's kind of, they've explored it, and it's probably not useful for like eradication in the wilds kind of environment. Yeah.
Do you know how they're currently using the snake-proof fencing? Like, there any possibility of like fencing off an area free from snakes? They could be confident in that, and then
Yeah,
I mean, birds will fly over them.
that's the main problem. Yeah. Yes. No, I'm realizing actually I should be repeating the question. Sorry, everybody online. That question was about, have they been doing snake-proofed areas, and would that work for, say maybe reintroducing birds? It's certainly being discussed. Yes, birds will probably fly over the fences most likely, so it's like may not be useful, but yes, that is one method that's been discussed, is like have some kind of area that's snake proof. I mean, one thing that we discovered not discovered is probably the wrong word, but it was emphasized relatively recently, is that we have an area that's currently has a 55-hectare area that's currently has experiments ongoing with eradication efforts. That's one-way closed, so snakes, I think, can come in but can't get out. It's one or the other. And there was a rather large typhoon a year or so ago, and it brought it right down. So there's also some question about, like, if particularly if, say, extreme weather events are more common. There are other methods that might work, other than the chain link fencing, but yeah, no, it's an idea for sure. It's expensive. It would be expensive to do probably, but it might be worth it. Eve.
If you absolutely had to choose which size class to throw your efforts into, which one would make the most sense? Because the large ones are reproducing, but then smaller classes are growing, so which one?
Yeah, Eve asked what size class, if you had to choose, would make the most sense. Definitely the extra large. There's been some studies that have looked at that a little bit, although their size class is a little different than mine, but it's close enough. Basically, the extra large ones can eat the most, and also they can reproduce the most likely. And so the small ones are going to be problematic, in the future, because they will grow, and it's a little bit unknown, like if we were to really kill off a lot of the larger ones, I'm not sure how quickly the small, I'm assuming a growth rate would be increased by doing that, but we don't know how much, we don't have any real idea of that. So, but we know for sure the extra large ones are killing the most stuff, or would be capable of killing the most stuff if we're reintroducing things, and they're reproducing the most. And so, yeah, pretty much gonna, but also it's hard to really target necessarily. We can target the ones that the methods are best for, but you can't really target beyond that. So yeah.
Got 3 comments on the.
Oh, okay. Brielle had her hand up too, but
So the problem of your ADS first estimation, is it because your detection probability is so low?
yeah, so the question was for ADS first, why was it so bad at estimating? And yes, well, that's true for everything, but basically there was very little effort. So low encounter rate plus only 3 weeks of visual surveys going on, even though I did have all those methods. I think I actually forgot to say this. All those methods were covering, quote unquote, the whole area. So visual surveys occurred throughout my fake 50 hectares, but because it was only 3 weeks, it was still just not that many times
As
So.
a sanity check, have you tried increasing that effort to see if it made it better? And like, can you find what that trade-off, like how much effort do you have to put in to actually get good estimates?
Yes, that's basically, so the question was, have I figured out how much effort is enough to sort of create good estimates? Short answer is no, and I would like to do that. I think that's probably gonna be where the next step that we're probably gonna be taking is to come up with a bunch of scenarios and like just generate some alternate strategies and just have a bunch more than two things to compare against each other. And so I would like to play with that a bit with those scenarios for sure. Have like effort ranging being one of the things that I'm varying.
Do you want to acknowledge the time so folks can
Oh, yeah.
right now at 1:30?
Go for it.
We're socially released from this obligation.
Thank you all.
Let's think going one more time.
Thank you. And then
there's-
I'm happy to answer,
Yeah.
I'm happy to answer all my questions.
Sarah says, thanks, Kelly. For those that are interested about the fences, kind of SAFS dissertation
Oh,
has
right.
lots of info on effectiveness of reintroduction in fence areas.
Yeah.
Also, 4 months is plenty.
Thanks, Sarah.
And then Noble Hendricks wants to know, I know, what is the mating strategy like for brown tree snakes?
Mm,
Any chance of being able to target like a breeding or mating aggregation?
yes. Okay, so the so there's a couple elements to that. There have been efforts for a while now to get pheromones to work as mating pheromones to work as a bait technique, and that has pretty much not worked so far. I'm not sure that it's fully been stopped efforts, but it's definitely not working. So the other thing is we have not managed to get them to reproduce in captivity. So we don't really know how they reproduce. But there's probably not aggregations. That seems pretty unlikely.
That'd be too convenient.
It would be way too convenient, yeah. Yeah, they seem to be pretty specially designed to be very difficult to manage. So.
The last one's from Stacy.
Oh,
Stacy says, nice work, Kelly. I particularly liked your visualizations to walk through the removal model. I also like those. Throughout this process, have there been specific pieces of info that you feel like would be worthwhile for specific future focus? Thinking about like value of info and future ground. Anything rise to the top for you? Wish list items.
Let's see. True thing. I mean, one of the things that I will be very shortly ground truthing that I feel a little bit naked about not already having is I will have actual removal data from an effort that I will eradication experiment on the ground that I will have all the effort for that I can like put into my model and see if what I'm getting out is similar to their actual data. So like that's a really important thing that feels like I'm excited to be able to do soon. And yeah, other than that, honestly, there's a lot of things. I think, yeah, certainly the way that I'm doing carrying capacity is pretty weird. So like having some ground truthing if in the future around that would be really valuable, because right now it's just kind of like it's just a static value that is not changing. So, and it's of course and that one is particularly fragile because if we're adding more prey items to an area, then that density dependence, it's really prey dependence mostly, and so it's going to change as you're adding potentially adding birds and stuff. So that one feels really important to focus on too.
Well, thank you for those fans that stayed online for your questions to be answered.
Thank you.
And thank you for attending. We'll see you next quarter.